Hi everyone, I am Maria Rodriguez, a master's student in the Katie Wachters Lab at the University of Wyoming. In my thesis, I'm currently working with Yellowstone cutthroat trout populations of the Teton River, and although I cannot join you in person, I'm very excited to share with you the work we are currently doing in Wyoming along with Friends of the Teton River. First of all, I want to introduce myself. I'm a biologist from Colombia, and coming from the highly biodiverse tropics, I'm very interested in understanding how biodiversity is formed, maintained, but also how we can conserve it. During my 8 months here, it has been a pleasure to learn so much about the biodiversity of the Western US, especially fish. The region's fish fauna is incredibly interesting and is giving us a lot of opportunities, but it also faces a lot of challenges. Jumping into what is happening with the Yellowstone cutthroat trout, you are well aware of the cultural and economic importance of these native subspecies in the West. Thus, the persisting population declines throughout its distribution are a major concern. We know that different human actions have been largely responsible for these declines, and they include activities such as the construction of artificial barriers that segment rivers, but also when we stock non-native species or even very different populations from the same species. The problem is that we have done these practices for a while, so we need to find ways that allow us to examine what have been their consequences on wild populations. Genetics, which in a broad sense is the study of the genes in the DNA and their variation, is very useful for this for two reasons. First, genetics is a valuable source of information to know the current state of populations. And second, it serves as a tool to infer how populations responded to events in the past. Starting with the first reason, you may be wondering why genetic diversity is even important. And to illustrate its relevance, I'm going to use the analogy of a toolbox. Let's imagine that we have our house, but unfortunately, a tree fell on it and it causes several damage. If we have a toolbox with only one tool, we know that we wouldn't be able to restore our house. However, if we have a toolbox that is very well equipped, we know that we would have the different tools we need to restore the damage. In other words, we would be prepared to respond to that external factor that was the tree. Genetic diversity works in a very similar way. Large populations that have a high genetic diversity are more likely to respond to external factors such as diseases, predators, and environmental change. In contrast, populations that are more genetically homogeneous have a hard time dealing with these factors, and they can even go extinct. Thus, we should care about the genetic diversity of wild populations because it is fundamental for their future persistence. Moving on to the second reason, genetics is very powerful because it allows us to infer how populations responded to events in the past. A parameter we are very interested in is population size, and it is because larger populations harbor a higher genetic variability. But the truth is, we lack records of population demography in the past because getting them requires prolonged and systematic fieldwork. Fortunately, genetics is a way to infer how population size has changed over time, because when both population expansions and declines occur, they leave specific signatures in the genome that we can detect. This is very useful in conservation because we are interested in understanding if population sizes have changed due to specific human actions. So after this brief background on the importance and use of genetics in conservation, I will describe the work we are currently doing with the Yellowstone cutthroat trout populations in the Teton River. We are particularly interested in understanding how genetic diversity is affected by barriers such as dams and the water streams, but also by the stocking of individuals coming from very different populations. As I already told you, genetic diversity could have decreased, either by leaving isolated populations of smaller sizes with barriers, or when we introduce genetically homogeneous stocks. Thus, our first objective is to characterize the levels of genetic diversity of populations, know how this variability is spatially distributed, and identify important tributaries for the maintenance of the Teton River population as a whole. For this, we are combining genetic data from approximately at 1,200 genetic samples with spatial data of the river and past stocking records. Our second objective is to understand how populations have been affected by the failed dam, which is a barrier that was built a century ago and left two disconnected river sections. We have preliminary evidence that populations upstream and downstream of the dam are genetically differentiated. So we want to explicitly test if the dam was responsible for this differentiation. To do this, we will use our available genetic data to know whether population declines occurred after the construction of this dam. This is going to be very relevant to understand what have been the effects of the failed dam, which will inform future conservation actions. The main take-home message of this is that genetics is a very valuable source of information and also an innovative tool that is giving us a novel perspective to address conservation problems. And this is very relevant for endangered species such as the cutthroat trout. Having said that, I am extremely thankful to Friends of the Teton River for the funding they are providing me to pursue my graduate studies in the University of Wyoming while I'm conducting this exciting project. My goal for my future is to apply genetics in conservation, and this is an incredible opportunity for me to do this. Thank you very much for listening to me and for your support that is making this project possible.